Okay, very good morning to everyone. I'm welcoming you to the AgriAddict. As you know, we have the teachers from different corner of the India and they are very experienced. So the thing you have to understand whatever the topic they have taken and whatever they are teaching and whatever the doubt you have to post or you have to put your doubt in the WhatsApp box in the slides they have given their contact number. Na? So at any time you can put your doubt in that WhatsApp box and we'll take all possible steps to clear all of your doubt. That's our prime job. So without any wasting any time, so I need to start today's topic. Today's topic is nervous system. Before that, my introduction. I am Pravu Malante. I'm currently pursuing my master degree in TNU Coimbatore. Okay. So earlier we have studied regarding the sense organ and today we are going to study regarding the insect nervous system. In insect nervous system what we have to mainly focus about what are the different terminology. Maximum time questions are coming from the terminology. What are the different terminology we have to be thorough about that. Then what are different type of neurons, what are the structure of neuron, what are the classification of the neurons, then what different type of nervous system like CNS, PNS, ANS that we have to also remember, and then nerve conduction. Nerve conduction we have to understand the theory, understand what is actually happening. Once you understand means you can able to do any type of question. Okay, because now the time question is not memory based, question is conceptual based, how much you grasp the knowledge. So that for that you have to be thorough understanding of that particular topic. Okay, so without delaying any time, so let's start the topic. So why first my question is why nervous system is required. So what is the reason of presence of nervous system in insect body? Look, in bacteria bacteria is the unicellular cellular organism. So a single cell is there, single inside a single cell one nucleus is there. So whatever the cellular mechanism of this cell, it is coordinated by the single cell. But in case of your multicellular organism, you can go to insect or you can go to any human being. So here they are all multicellular organism. Here small single cell combined to form a group of cell, and again this group of cell combine to form the tissue, then tissue from the organ, then organ from the organ system. So there is a whole means complex mechanism happening in our body. To control that one, definitely some coordinated system in the body must be there, that is called as your nervous system. Okay, then another peculiarity and very fundamental thing about the insect nervous system is it is present. If you look, our nervous system is present at a dorsal side. Our spinal cord and this are all spinal cord portion and the brain portion. This is all present in the dorsal side. But in case of an insect, it is present in the ventral side. That you have to remember. In case of an insect, the nervous system is present in the ventral side. That is why whenever you are doing practical to dissect the nervous system of the insect body, you have to start from the dorsal side. Okay, because nervous system is present in the ventral side. So if you look at the mechanism of the nerve conduction, we have already studied there are different type of sensilla present in the insect body, like for an example chordotonal sensilla, camponiform sensilla, trichoid sensilla. There are different type of sensilla present in the environment, in the insect body. And now this sensilla mainly receive the signals that from the external environment, whatever the environment surrounding the insect body. From there itself, these sensilla are taking the signals, impulse, stimuli, whatever you want to say. And this stimuli, whatever the impulse they are taking, must be taken into the action. So that is done by the nervous system. Nervous system process that stimuli, whatever the stimuli that is, and then it is command to the effector organ. Effector organ may be muscles or any tissue or any organ, you can say organ. For an example, suppose an insect is there at certain distance, some food source is there, and from that food source the volatile substances are coming. Now this volatile substances acts as the stimuli or impulse. Now these volatile substances are conveyed by the different sensilla present in the insect antenna. So once this stimuli is conveyed by the insect antenna, it will convey to the nervous system. Now nervous system will command to the effectory organ. Effectory excretory organ. For an example, mouth, you can say leg. Now it will command the mouth and leg to reach up to the food and then take the food. Okay, this is the whole process how it's happening. So if you look at the nervous system of the insect, it is divided into two parts. One is your nervous part, another is your non-nervous part. So in nervous part, the main cell is your neuron. Neuron is the cell, means the nerve cell, you can say. The main cell is the neuron, structural and functional unit of the nervous system. Many times it is asked in the question, what is the structural and functional unit of the nervous system? That is your neuron. Neuron is the structural and functional unit of the nervous system. Apart from neuron, in the non-nervous part, there are neural lamella is there, neuroglial cell is there, and neurologia is there. There are different type of cells are there, which main function generally they are cover the neuron and they provide the nutrition to the neurons. Okay, so nervous system, if you look, it is divided into two parts. One is your nervous part, which mainly consists of your neuron. Another is your non-nervous part, that is consist of different type of cell. That is the supportive cell that is mainly provide the nutrition to the neurons. Okay, so if you look at the structure of the neuron, this is the whole structure of the neuron. We have already we read regarding the structure of the neuron, lots 2 and all, but we have to little thorough regarding the terminology. So if you look at the structure of neuron, there are generally three type of part is present. One is your dendron, first one is your cell body, second one is your dendron, and third one is your axon. Okay, so look, this is the structure of the neuron. In the structure of the neuron, where nucleus is present, look, this one is the nucleus. Where nucleus is present, here this one is the nucleus. Where nucleus is present, that is called as your cell body. There are different other name for the cell body also. You should not be confused in that name. You have to remember. Cell body is also called as your soma. It is also called as your perikaryon. It is also called as your neurocytes. There are three. There are four. Means four name has same meaning. Cell body, that is also called as your soma, that is also called as your perikaryon, and that is also called as your neurocytes. Okay, so the structure of the neuron where nucleus is present, that is called as your cell body. From this part, there are several minute protoplasmic outgrowth generally found. The small protoplasmic outgrowth, these are the small protoplasmic outgrowth. The small protoplasmic outgrowth is called as your dendron, and the longest protoplasmic outgrowth is called as your axon. Okay, so understood, in this part of the neuron where nucleus is present and surrounding the nucleus the protoplasm is present, that is called as your cell body. Cell body has also different name, that is soma, perikaryon, neurocytes. Okay, and from that part there are several protoplasmic outgrowth or extension generally found, and the small protoplasmic extension is called as your dendron, and the long protoplasmic extension is called as your axon. Okay, then throughout the length of the axon several branches are present. Look, these are the branches. From the length of the axon, several branches are present. These branches is called as your collateral or axon collateral, whatever you want to say. This branches is called as your collateral, and the this tip of all extension, whether it may be present in axon, it may be present in dendron, the tip one is called as your terminal arborization. The tip one is what you call, that is called as your terminal arborization. And the portion from where axon arises, from the cell body, you can see this neck like portion that is called as your axon. This is called as your axon hillock, 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 that is called as your axon hillock. So there are so many terminology you have to remember. So neuron is the neuron is the structural and functional unit of the nervous system. Neuron is divided into three parts. First one is you can say cell body. Its other name is soma, perikaryon, or neurocytes. The portion of the neuron where nucleus is present surrounding by the protoplasm, that is called as a neuron. Then from the, that is called as your cell body. I'm sorry, that is called as your cell body. From the cell body, there are too many protoplasmic extension are coming out. The small protoplasmic extension is called as your dendron, and the long protoplasmic extension is called as your axon. The portion from where axon arises from the cell body, that is called as your axon hillock. In the length of the axon, there are so many branches are present, that is called as your collateral or axon collateral, you can say. And the terminal portion of the each of the branches or each of the tip, you can say, that is called as your terminal arborization. Terminal arborization may be the terminal portion of the dendron, may be the terminal portion of the collateral, also may be the terminal portion of the axon. You no need to confuse. This is the schematic diagram. You have to keep this diagram in mind. This is very important. If it is the structure of the neurons, the cell body, here nucleus is present. It is also called as your perikaryon, cell body, soma, and neurocytes. From where this axon arises, that is called as your axon hillock. And from the axon, there are the branches is present, that is called as your axon collateral. Okay. And from the cell body, small minute like extension arises, that is called as your dendrons. And the tip of the each branches or tip of the structure, that is called as your terminal arborization. Okay, then also this terminal arborization can be present either in axon or axon collateral or dendron. This is the schematic diagram. You just have to remember this diagram. Okay, then many times we heard a term, that is your ganglia. In case of particular insect, we are hearing a term ganglia. Look, ganglia is different and glial cell, you may be confused in between them. Glial cell is also different. From the very first slide, I have told you insect nervous system is divided into two parts. One is your nervous part, which mainly consists of neuron. Another is your non-nervous part. There itself glial cell is coming, and the main function is of the glial cell is the providing nourishment to the neurons. So similarly, ganglia is somehow it's very different term, and it is the group of neuron. When the group of neuron present at a place together, that is called as your ganglia. If a group of axon is present, that is called as your neuropile. So understood? If group of neuron is present, then that is called as your ganglia. If group of axon is present, that is called as your neuropile. Then, if you see if you categorize the neuron, there are two bases. One is your structural basis, another is your functional basis. On structural basis, neuron is divided into unipolar, pseudo-unipolar, bipolar, and multipolar. Okay, so we have to know on which basis we are categorizing, on which structure basis we are categorizing. It's all on the basis of presence of axon and dendron. Axon and dendron. Oh, look, this is the cell body. Here only one axon, this one is the axon, only one axon is present, and this one is the axon collateral. But from the cell body itself, there is no dendron. There is no dendron. So that is called unipolar. That means in unipolar, dendron is absent. Only one axon is present, but some dendron can be found in the axon collateral. That doesn't matter. We have to check whether dendron arises from the cell body or soma. Okay, that we have to check. If dendron is not present, not arises from the cell body or soma, we can say that is your unipolar. That mainly consists of one axon. Similarly, this is the structure of another unipolar neuron, where look, this is the cell body. There is no dendron is present, only one axon is present, but it's looking like bipolar. This is what? This is the axon collateral. This is very simple. Axon collateral. You may see this is the dendron. This is the axon. We can say it is the bipolar. No, you have to check whether dendron arises from the cell body or soma or not. Dendron may be present in the axon collateral, but you have to check whether it is arises from the cell body or soma or not. If it is not present in the cell body or soma, and only axon is present in the neuron, you can say it is the unipolar neuron. Then another, if you to take this one is the bipolar neuron. Look, this is the cell body. From the cell body itself, the dendron is there. From the cell body itself, the dendron arises, and one axon is present. So if one dendron and one axon is present, that is called as your bipolar. Look, this one is the cell body. From the cell body, there are multiple number of dendron arises and one axon is present. If multiple number of dendron is present and one axon is present, that is called as your multipolar. Understood? So on what now you cleared on what basis we are categorizing? We have to check for the presence of axon and we have to check for the presence of dendron. That dendron particularized, it should be arise from the cell body. If it is not arise from the cell body and arise from the other part, like from axon collateral or tip is also called as a dendron. If it is arise from there, then we can take into the categorized consideration. Okay, so if from the cell body or soma there is no dendron is present, only one axon is present, that is called as your unipolar. If from the cell body only one dendron is present and one axon is present, that is called as your bipolar. If from the cell body numerous number of dendron is present and only one axon is present, that is called as your multipolar. I'll write it. I'll write it. Where is this? This is the, you take it, this is suppose unipolar, then we have bipolar, then we have multipolar. This is dendron, this is your axon. So sorry, sorry, this is not, I will write unipolar, bipolar and multipolar, and this is your dendron, this is your axon. Look, in unipolar there is dendron is 0, dendron is 0, axon is 1. In bipolar, dendron is 1, axon is 1. In multipolar, dendron is n number and axon is also 1. So you have to see in every case this the only one axon is present, but we have to check for the presence of the dendron. But in case of your multipolar, also numerous number of axon may be present. As the numerous number of group of axon is present, that's why you are using the term neuropile. Previously we see na, we are using the term neuropile, so that can be happen in multipolar condition. Okay, and what is the pseudo unipolar? It is looking like one dendron is present, but that one dendron is not arises from the cell body, it may be arises from the axon collateral. So it is similar like bipolar, but we can consider it as the bipolar. This is again unipolar type because there is no dendron arises from the cell body. Then this is another classification of the neuron where this is on the basis of function, what the neuron is performing. So on that basis, we are generally classifying the neuron into three categories. One is your afferent, the other name is sensory, another is your efferent. Sorry, so this is E. This is E. Afferent, then efferent. E F R E and efferent. The other name is effector or motor. Three name. This is my wrong. This is my mistake. This is not afferent. This is efferent or effector or motor. Another is an intentional or association or association type of neurons. So three type of neurons on the basis of function, because neuron may perform different type of function. We will see that function. So on the basis of function, three type of neuron: afferent neuron, which is also called as your sensory neuron; efferent neuron, that is also called as your motor neuron or effectory neuron; and intentional type of neuron or association type of neurons. So as the name suggesting, afferent neurons or afferent neuron or sensory neuron, it will transmit the impulse from the sense organ to the central nervous system. So from the sense organ to the central nervous system, as it is transmitting the impulse, that's why the name is sensory. And maximum type of sensory neurons are either bipolar or multipolar type. Okay, means one dendron must be there. If it is multipolar, numerous number of dendron and numerous number of axon will be present. Then from the central nervous system, by processing this impulse, whatever the command that is given by central nervous system, it will perceive to the effector organ through the motor neurons or efferent type of neurons. So maximum type of motor neurons are unipolar. So in between this central nervous system, there is another type of neurons are present, that is called as your internensal type of neurons are present, that is present only in the central nervous system, that is bridging or connecting the afferent neuron and efferent neuron. That is also called as your internensal or association type of neurons, and that is also unipolar type of neurons. So you understood those neurons which transmit the impulse from the sense organ or sensilla to the central nervous system, that is called as your afferent or sensory neurons, that is maybe bipolar or multipolar in nature. Those neurons that transmit the impulse from central nervous system, transmit the commands, you can say commands from the central nervous system to the effectory organ, maybe muscles or some other organ system or tissue, that is called as your efferent neurons or motor neurons, and these are maximum unipolar in nature, and there are some certain unipolar neurons that is present mainly in the central nervous system, that is bridging or connecting the afferent neuron as well as your efferent neuron, that is called as your internecine type of neuron system, internecine or association type of neurons. Okay, then see another some word. Yeah, if you see the nervous system, if you dissect the nervous system, the neurons are surrounded by various non-nervous cells, as we have discussed earlier, and then all this structure is surrounded by a thin sheet of tissue that is called as a neural lamella. That is called as a neural lamella, and the other name of the neural lamella that is called as a neurilemma. And the other name of the neurilemma that is also is your perineurium. So this three name you have to remember. The if the question been asked, the whole nervous system is surrounded by whom, you have to say it may be neural lamella, neurilemma, or perineurium. It has very important function. In human we are saying it, the perineurium has the function that is your blood brain barrier. But in insect also blood is not present. Suppose this one is the nervous system, and surrounding the nervous system the structure that is present, that is called as a hemolymph, and in hemolymph there are several ions are present. For an example, I can say sodium ions are there, potassium ions are there, several ions are present, and this perineurium mainly guides the entry and exit of the ions inside the nervous system, particularly this potassium ions. So this is also called as your blood brain barrier in insect also, hemolymph brain barrier in insect also. So you have to remember that one. Three name for that: neural lamina, neurilemma, and perineurium. All are same, and generally it is covering the nervous system, and it mainly regulates the exchange of ion, particularly potassium, in and out of the nervous system. Then another term, what is neurolemma? This is neurilemma. There is the small sheet of structure that is covering the only portion of the axon, that is called as your neurolemma. So you have to see the question, what the question is asking, whether this structure, whether that small thin sheet is surrounding the whole nervous system or the axon. If it is only the axon, then you have to answer the neurolemma. If it is surrounding the whole nervous system, then you have to answer that is your neurilemma or neural lamella or perineurium, which is also called as your hemolymph brain barrier. Then some of the important points, if you see the normal diameter, it may be asked, it's memory based question, normal diameter of the nervous system, that is your 5 micrometer. But there are several if you see in the central nervous system, several neurons are there, where axon length exceeds more than 60 micrometer, axon diameter exceeds more than 60 micrometer, that is called as your giant fiber. So mainly if this is the sensory, this is the motor, and this is the central nervous system. Inside the central nervous system there is the internecine type of neurons is present. This internecine type of neurons generally very long, its diameter can exceed more than 60 micrometer itself, and these are also called as a giant fiber. Why this diameter is too long? Because they, for the rapid conduction of the nerve impulse. Okay, then another thing is Nissel's granules. Nissel's granules generally, if you look at the structure, this is the nervous system, this is the cell body, and this is the dendrites. In the dendrites, for the rapid conduction, definitely it's need the protein. So for the protein production in the dendrites, that is raw endoplasmic reticulum is present, and this raw endoplasmic reticulum structure that is present in the dendrites is called as your Nissel's granules. Okay, so Nissel's granules is the structure. Generally, it is present in the dendrites, and it has the raw endoplasmic reticulum, which mainly produce the protein, and this protein is very essential for the conduction of the nerve impulse. Okay, so generally we study what are the ganglia. Ganglia are the group of the neurons. Okay, so these ganglia are present like this. Okay, so these ganglia are longitudinally connected. or transversely connected. This term many times it comes in the exam, so please you remember. This transverse connection is called as your commissures. This transverse connection is called as your commissures, whereas this longitudinal connection is called as your connectives. So there are two type of connection. One is your connectives, another is your commissures. The transverse connection is called as your commissures and the longitudinal connection is called as your connectives. Then another very peculiar characteristic of insect nervous system is the absence of motor end plate. What is motor end plate? Generally, it is the structure. It is present beyond the axon. When the axon is connected with the muscles, in case of your human or in case of your higher organism, that structure is present. That is called as a motor end plate. Its main function is to regulate the passage of the nerve impulses. So this motor end plate generally absent in case of your insect. So these are some memory based point you have to remember that insect normal diameter of the insect nervous system is 5 micrometer, but some internal neurons or association type of neurons, its diameter can exceed 60 micrometer or more than that for the rapid conduction of the nerve impulse. So that is called as your giant fiber. Then in dendrites, micelle granule is present. Micelle granules mainly contains the raw endoplasmic reticulum. It may be asked what is the position of the micelle granules. You have to answer that is dendrites. So in dendrites, micelle granules is present. It mainly consists of your raw endoplasmic reticulum, and this raw endoplasmic reticulum mainly concerned with the production of the proteins. And if you see the connection in between the ganglia, there are two type of connection is present. One is your longitudinal connection, which is called as your connectives, and another is your transverse connection, which is called as your commissures. Okay. Then another very peculiar term we are studying, that is your synapse, and synapse has highly importance during the conduction of the nerve. So we will again study the synapse in some later slides, but before that just take some glimpses. Suppose this is the nervous system. This is one of the nervous system. Okay, this is cell body and dendrite, and this is the axon, and axon just terminates here. From here arises another nervous system. The gap between this, that is called as your synapse. This may be axon, this may be dendron. This is the axon, this is the dendron, or this may be the dendron, this may be the dendron, or this may be the dendron, this may be the axon. Okay, so we can say the gap between the two neuron, that is called as your synapse. Okay, in this gap it may be the axon. The gap between two axon can be called as the synapse, or the gap between an axon and another dendron, that is called as your also called as your synapse. So then how we'll say, look, if it is the unipolar neuron, then definitely here there is no dendron. So there's the gap between the axon and another axon, that is called as your synapse. If it is the bipolar, here one axon is there, one dendron is there. So that is also called as your synapse. So how you will remember this? The gap between two neurons, whatever that may be, axon, dendron, or anything, the gap between the two neurons, that is called as your synapse. Many times it has been asked, what is the size of synapse? The size of the synapse is 100 angstrom. 100 angstrom. 1 angstrom is 10 to the power minus 10 meter. So from this, it may be asked in the angstrom, it may be asked in the micrometer, it may be asked in the nanometer. Accordingly, you have to convert, like 1 angstrom, sorry, 1 angstrom is equal to 10 to the power minus 10 meter. So this is the basic unit. From that you can convert into nanometer. 1 nanometer is 10 to the power minus 9 meter. 1 micrometer is 10 to the power minus 6 meter. So you don't expect the question answer will or option will only come in the form of angstrom. It may be common in the form of any unit. So you have to be prepared for that. And the synapse can be divided into axosomatic, axodendritic, and axoaxonic. What is this? I have already told you. This is the gap between two neurons. If for an example, this upper neurons is bipolar in nature, means the end is the axon. The lower neurons is the unipolar in nature. That means the starting one is the cell body or soma. Definitely, it's the gap between the axon and soma. That's why it is called as an axosomatic type of synapse. Then axodendritic. Suppose this one is the bipolar neuron and this one is the again bipolar neuron. Then definitely it will be the axon and it will be the what? it will be the dendron. So definitely that is this type of synapse is called as an axodendritic type. Then axonic type. This is the bipolar neuron. This is the bipolar neuron, but it is reversely arranged. So this is the gap between axon and axon. So that is called as an axonic type. So understood. So for that only you have to remember it is the gap between two neurons, whatever it may be. Cell body, gap between the axon cell body, gap between the axon, or gap between the axon and dendrites. Then this is the general structure of the nervous system, where we studied about the neurons, what are the different part of neurons, what are other structure that is associated with the neurons, and we classify the neurons. Then we'll come to the insect nervous system. If we'll see in the insect nervous system, generally like human, the insect nervous system is also categorized into three parts: central nervous system, peripheral nervous system, and ANS, that we'll see, sympathetic nervous systems. Okay, so this is the central nervous system. It is generally divided into brain, another is just subesophageal ganglia, another is the ventral ganglia. Okay, so brain, subesophageal, central nervous system is generally divided into brain, subesophageal ganglia, and each are ventral ganglia. Then in brain, generally 6 segment is there. We already studied insect brain consists of 6 segments. Each segment consists of your one neuromere. Each segment consists of one neuromere. So what is neuromere? Neuromere means it is the group of ganglia, you can say. It is the group of means nervous, non-nervous part all combined and form a group that is called as your neuromere. Each segment consists of one neuromere. So first three segment must have 3 neuromere, and these 3 neuromere will form as your brain. This 3 neuromere is formed as your brain. Latter 3 neuromere that is formed as your subesophageal ganglia. Understood? So brain has 6 neuromere. 6 neuromere. First three, last three. First 3 neuromere that is formed as your brain, and last 3 neuromere that is formed as your subesophageal ganglia. And then you have to remember again, brain is divided into protocerebrum, deuterocerebrum, and tritocerebrum. Maximum time it has been asked in the question, for an example, the nerve into the antenna is coming from which part? The nerve into the mandible is coming from the which part? You have to remember that. From protocerebrum, the nerve has generally passes to the compound eye and ocelli. That means optic lobe. Optic lobe must be present in the protocerebrum. From deuterocerebrum, the nerve is passing to the antenna. That means antennal lobe present in the deuterocerebrum. From tritocerebrum, the nerve is only passes to the upper part of the mouth, that is called as a labrum. Okay, so what you have to remember, brain generally consists of, whatever the insect head we are saying, head consists of 6 segments. Out of these 6 segments, first 3 segments generally constitute the brain, last 3 segments form the subesophageal ganglia. This brain is again divided into 3 categories: protocerebrum, which generally conveys the nerve to the compound eye and ocelli. Then deuterocerebrum, it conveys the nerve to the antenna. Then tritocerebrum, it conveys the nerve to the labrum. Then subesophageal ganglia. Subesophageal ganglia formed by last 3 cephalic neuromere. Cephalic means brain. Cephalic, the word we are using whenever the brain is there, we are using the cephalic word for that. So last 3 cephalic neuromere generally form the subesophageal ganglia, and it is generally convey the signals from the mouth part, salivary gland, and neck. Then the other neuromere that is present in the thoracic region and abdominal region, that is formed as your ventral ganglia. In thoracic region there are 3 segments is there, as I have already told you in is. So this ventral ganglia mainly consists of whatever the neuromere those are present in the thoracic region as well as in the abdominal region. As I have told you, you have to remember one basic point, that in each segment of the insect there is only one neuromere is present. We have discussed in head consists of 6 segments. So 6 neuromere that constitute 3 of the first neuromere that constitute the brain, and 3 of the last neuromere that is constitute the subesophageal ganglia. Similarly, whatever the neurons those are present in the thorax and abdomen, it will form the ventral ganglia. So thorax has the 3 segments, so definitely this consists of 3 thoracic neuromere. So thoracic ganglia consists of 3 thoracic neuromere, and it is generally passes the nerve, whatever the structure those are present in the thorax, that means the legs and as well as the wings. Abdominal, it consists of 11 segments. So generally 11 neurovere is present, but in insects generally these 11 neurovere degenerate and it is converted into 8 pair of neurons, and this 8 pair of neurons, it will provide the that nerve to the spiracles. So you have to remember, this is not 8 pairs, this is 8 neurons. Sorry, it is not 8 pair, this is my fault, I'm sorry for that. This is not 8 pair, this is only 8 neurons. So insect head consists of 6 segments, 6 segment, 3 brain, 3 subesophageal ganglion, 3 thoracic segment from the 3 thoracic ganglion, and 11 abdominal segment. It may be present in some Thysanura. All the segment, abdominal segment consist of your neuromeres, but in other insects generally these 11 neuromere degenerate and it is from the 8 neuromeres, out of which last neuromeres generally enlarge. This the last 9, 10 and 11 neuromeres that combine to form the last neuromeres. So that is happening in maximum insect. Okay, so 3 plus 3 plus 3, 9 plus 8, total 17 neuromeres present in case of a normal insect. But if you look at the theory, means look in some that primitive insect like Thysanura, there are in all segments like 3 plus 3 plus 3, 9 plus 11, 20 neuromeres is present in case of an insect. If it has been asked you in the question how many neuromeres is present in case of an insect, then you have to particularly answer for the 17. If it is only asked for the Thysanura, then you have to answer for the 20 neuromeres is present, because in Thysanura all abdominal segment consist of your neuromeres. Then there are some important points regarding the central nervous system. You have to remember that one. In case of your desert locust, that metathoracic ganglia, mesothoracic ganglia. Abdomen has 3 thoracic ganglia: prothoracic ganglia, mesothoracic ganglia, metathoracic ganglia. Mesothoracic ganglia, it conveys the nervous to the forelegs and mid legs, and forewings, and various type of chordotonal organ. Chordotonal organ we have read in the sensilla. It is the different type of sensilla. It is a type of mechanoreceptors. So in locus, the mesothoracic ganglia, it supplies the nerve to this part of the body, and metathoracic ganglia, it is the compound ganglia. Why I am saying it is the compound ganglia means metathoracic ganglia. This is the metathoracic ganglia along with first or second abdominal ganglia. It form a compound ganglia. So it is not only the metathoracic ganglia, it is the combination of the metathoracic ganglia with first and second abdominal ganglia. This compound ganglia, it will convey the nerve to the hind wings, hind leg, and spiracle. Okay, then largest number of the abdominal ganglia, as I have told earlier, it is present in case of your Thysanoptera and male pulex species. Pulex, it should be italicized. Again, it's my fault. So whenever you are writing a scientific name, you have to be careful. It should be italicized. So it is 11 number of that is abdominal ganglia. It is present in case of your Thysanura and pulex, male pulex, which is the Siphanoptera. So you have to remember them. Then terminal ganglia, in many cases we have seen terminal, in many insect we have seen terminal ganglia is there. As I have told you, there are 8 ganglia is there. That means the terminal ganglia is the fusion of the 9, 10, 11 and 8 abdominal, 8, 9, 10 and 11 abdominal ganglia combines to form a large ganglia that is called as your terminal ganglia. Then segmental autonomy in insect. Why we are saying in segmental autonomy? Because in each segment one neuromere or one ganglia is present. That's why we are saying it's segmental autonomy. Each segment control its own function. Whatever the ganglia that is present in the prothoracic segment, it is control the function of the prothoracic region. Whatever the ganglia present in mesothoracic segment, it is control the function of the mesothoracic segment. As each segment control its own function, that is called as your segmental autonomy, which is found in case of your insect. Then regional specialization. What is regional specialization? I can say that is we studied that is the compound ganglia. For an example, in some insect that is Chironomus and Schistosera gregaria. That's why we studied in locust. Schistosera gregaria is the scientific name of the desert locust. The compound ganglia is present. It's the regionally present, means it is the metathoracic ganglia in combination of the first and second abdominal segment. So that is also you call as your compound ganglia. So compound ganglia is also present in case of a Chironomus and desert locust. That is called, why we are calling it regional specialization, because it is the combination of the all ganglia, combination of different ganglia in a particular region. In case of locust, this is the metathoracic region. Okay, all 3 thoracic ganglia that is present in case of your straciomia. Trachea is a dipteran fly. Here in this case, both all three type of ganglia, prothoracic, mesothoracic, metathoracic ganglia, that is fused. All thoracic ganglia and abdominal ganglia. This is very interesting case. All the three thoracic ganglia and 11 abdominal ganglia, that is fused in case of your Disterchus koenigi and musca. In case of your Musca domestica, okay, musca is very commonly used fly. Musca domestica and Disterchus koenigi, in that case, all the thoracic and abdominal ganglia are fused. Then thoracic ganglia is fused with the subesophageal ganglia in plant lice and scale insect. In plant lice and scale insect, generally thoracic ganglia fused with the subesophageal ganglia. So you remember this modification: thoracic ganglia fused with first or second abdominal ganglia, that is the metathoracic ganglia or compound ganglia or regional specialization found in case of your desert locust and chironomous. Thoracic ganglia fused with all abdominal ganglia, that is found in case of your musca or Disterchus. Then thoracic ganglia fused with subesophageal ganglia, it is found in case of your plant lice or scale insect. Then another nervous system, that is your visceral nervous system, which is also called as your sympathetic nervous system. The visceral nervous system, generally it is three type. One is your esophageal or stomatogastric nervous system, which is also called as your anterior nervous system, and it is consists this type of ganglia: frontal ganglia, ingluvial ganglia, hypocerebral ganglia, and esophageal ganglia. You have to remember the frontal ganglia, that is mainly responsible for the controlled swallowing. Particularly, it's in every insect is responsible for the controlling of the swallowing of the food. In ingluvial ganglia, that is responsible for the controlling of the peristalsis. You remember this too. For the swallowing, frontal ganglia is associated. For the peristaltic movement, means after swallowing, the food will pass through the different part of the digestive system. Na? For that only ingluvial ganglia is responsible. Generally, in case of your locust, the frontal ganglia also regulate the corpora caudicum to secrete various functions. Corpora caudicum mainly responsible for the secrete that are various secretions, you can say various hormones. This is one of the endocrine gland mainly associated with the heart function. So this all hormone secretion function, it is regulated by the frontal ganglia in locust. Then hypocerebral ganglia, which is associated with the subesophageal ganglia and is regulate the function of the brain and subesophageal ganglia. Then esophageal ganglia. Esophageal, the other name of the esophageal ganglia, that is called as your corpora caudicum, which regulate the functions of the heart. So you understood. The first one is the esophageal or stomatostatic or anterior sympathetic nervous system. That is consist of four type of ganglia. Frontal ganglia, that is mainly associated with the swallowing of the food. Then ingluvial ganglia, which is mainly associated with the controlling peristalsis. Then hypocerebral ganglia, that is associated with the functioning of the brain and esophageal ganglia. Then esophageal ganglia, that is called as your corpus caudicum. So you remember there are three type of three means by combination of the esophagus. First one is the brain, which is also called as a supraesophageal ganglia. I forgot to tell you. Remember the other name of the brain, that is your supraesophageal, supraesophageal, esophageal. Supraesophageal ganglia, that is called as your brain. Then below that is your subesophageal ganglia is present. Then below that only esophageal ganglia is present. Supraesophageal ganglia is called as your brain. Subesophageal ganglia is your subesophageal ganglia. There is no other name for this. And esophageal ganglia is called as your corpora caudicum. Okay. Then another type of ventral sympathetic, another type of visceral nervous system is ventral sympathetic nervous system, and ventral sympathetic nervous system is generally regulate the function of the whatever the function of the whatever the spiracles, legs, and other tissues and organs those are present in the abdomen and thoracic region. Then another type of nervous system is the peripheral nervous system. The peripheral nervous system that mainly consists of the nerve that is arises from the central nervous system and sympathetic nervous system. Here it is written that is PNS. No, it is not PNS. This is SNS. So the peripheral nervous system consists of the nerve from the central nervous system and sympathetic nervous system. It mainly associated with the nerves. Those are mainly reaching to the either in the muscles or associated with the sensilla. So you understood there are three type of nervous system: central nervous system, and from central nervous system there are brain is subesophageal ganglia and ventral ganglia is present, and to which part they are supplying the nerve, also you have to remember that one. Then another is visceral or sympathetic nervous system. It is consist of your that is a ventral nervous system and esophageal or stomatogastric nervous system and caudal nervous system. Caudal nervous system, that is nothing, it supplies the nerve at different tail regions, spiracles and abdomen, with various abdominal region up to various abdominal region, generally it is supplies the nerve. Then peripheral nervous system, it mainly consists of the nerves that is arises from the central nervous system and sympathetic nervous system. Then there are some important point you have to be thorough regarding this. First one is the holy sheath. Holy sheath and perineurium, I have told, or in together, I have already told the nervous system or the neuron is surrounded by a thin sheet of cover. This thin sheet of cover is called as your neural lamella or neurinema or perineurium, which is also called as a blood-brain barrier, that is mainly associated for the potassium ion regulation. And this potassium ion regulation is done by a particular sheath present in the this perineurium, that is called as a holy sheath. So holy sheath is a minute sheath that is associated with the perineurium, that is mainly regulate the potassium ion. In the question, perineurium is there and holy sheath is there, more specifically you have to choose the perineurium. If both option is there, also you have to choose the both. If holy sheath is not there, you have to only go for the perineurium. Then corpora pedunculata. What are the corpora pedunculata? Corpora pedunculata, it's generally, I will tell it in the endocrine system. It mainly associated with the endocrine system. I will tell it in the endocrine system. Then glial cell, this is the non-nervous cell and it is provide the nutrition to the neurons. Then swan cells, we have already studied regarding the Nissel's granules. Nissel's granule, then that is mainly it is surrounding it is present in dendrites and it is consists of the raw endoplasmic reticulum that is secretes the protein. So swan cells mainly consist of the Nissel's granules and it is surrounding or covering the neurons, that is present everywhere. Then trophospongia. Trophospongia is a part of the nervous system when it is attached with the prototrophos. This is the normal cell and this is the neurons, and the portion of the neurons when it is attached with the protoplasm of a particular cell, that portion is called as a trophospongia. So trophospongia is the portion of the neuron where it is attached with the protoplasm of another cell, that is called as a trophospongia. So you understood these terms. So before going to the impulse conduction, I thought you may get some confusion, my own perception, you may get some confusion in that nervous system. In nervous system, that central nervous system you have thoroughly cleared, but in sympathetic nervous system, let me tell you another time, it is divided into three categories. One is your ventral nervous system, caudal nervous system and stomatogastric nervous system. Stomatogastric nervous system consists of different ganglia: frontal ganglia, ingluvial ganglia, hypocerebral ganglia and esophageal ganglia. Esophageal ganglia is called as a corpora cardiaca, that mainly regulates different type of heart function. Then ventral nervous system, it supplies the nerve various thoracic organs, and caudal nervous system, it supplies the nerve those organs those are present in the tail region or spiracles of the abdomen. So no need to get confused. And peripheral nervous system, it mainly consists of the nerves that mainly arises from the central nervous system and sympathetic nervous system. After that, we saw some of the important terms like your holy sheath, which is mainly responsible for the potassium ion regulation. Trophospongia, the part of the neurons where it associated with the protoplasm of other cells, that you have to take care. Then impulse conduction, how these impulses are conducted. So before that, you have to remember the stimuli. What is stimuli? Stimuli is the any change that can causes the alternation of the metabolic activity in the living cell, that is called as your stimuli. Inside the living cell, there are too many chemical reaction is happening. If any particular change is happening in that metabolic activity through some impulses, that is called as a stimuli. Then this impulses is passes from one cell to another cell. How? Because this protoplasm are connected. Through this protoplasm, these changes are processed from one cell to another cell, and there is the series of chain happening, that is called as your conductivity. Okay. Then another term is the recovery period. That is your time required. Whatever the changes, for an example, you gave a stimuli to this cell. So due to due after getting the stimuli, this metabolic activity of the cell is changed. Then whatever the time required to convert into the original state, again convert into the original metabolic processes, that is called as your recovery period. So time required to recover the original condition is called as your recovery period. So if you look at the nerve conduction, there are two type of nerve conduction is general there. One is your axonic conduction, another is your synaptic conduction. Impulse conducted through the length of axon, and after reaching the end of the axon, it passes from one axon to another neuron through the synapse. Okay, so this axonic conduction are generally electrical in nature, and synaptic conduction are biochemical in nature. How? I'll tell you. You first understand. You first listen to me and understand how it's happening. So you just forget about this charge, and this is the this you think this is the simple neurons. Okay, so in normal condition, when there is no stimulus there inside the neurons, you know there are several ions that are present inside the neurons, and in the length of the neurons there are several ionic channel is present. One of the most important ionic channel is your sodium potassium ionic channel. What we have to be studied in the nerve conduction point of view. Also chloride ionic channel, this is also very important. So in this length of the axon, several ionic channels are present. So in the resting stage inside, there are more number of potassium ion is present, and outside the neuron there are more number of sodium ion is present. If you look at the chemistry, sodium ion is more stronger as compared to the potassium ion. That's why it's outside the neuron there is positive charge, whereas inside the neuron there is negative charge. This is the condition in the resting stage, I can say, in the resting stage, where there is no impulse, no external stimuli is happening. Means in the resting stage inside the neuron there are more number of potassium ion is present, and outside the neuron there is more number of sodium ion is present. So outside there is positive charge, inside there is negative charge. When an impulse or when an external stimuli conveys, there causes the opening of certain channels, opening of the sodium potassium channels. Due to opening of the sodium potassium channels, the potassium will go out. As more number of potassium present inside, it will definitely tends to go out, and the sodium will come in. So due to accumulation of the more number of the sodium inside, the inside now becomes the positive charge and outside becomes the negative charge. So this condition is called as your depolarization, and that is called as your action potential. So now this depolarization, now this it will pass through conductivity. Conductivity we studied the changes that will pass from protoplasm to protoplasm. Now this series of depolarization, it is passed from throughout the length of the neurons, and it will terminate at the end of the neurons, and from the end of the neurons to another neurons, the generally the axonic transmissions happen through biochemical transmissions. Okay, so this is the just common point we have to remember on this. So there are some common terms we have to remember for that. One is your resting potential. Resting potential, which is all the other name for the resting potential is membrane potential. What I have already explained to you, in the resting condition, there are more number of potassium ion inside, and sorry, there are more number of potassium ion inside, and the outside there is more number of sodium ion is there. That's why the outside is positively charged and inside is negative charge. That condition is called as your resting potential, and if you measure the resting potential, it will come as your minus 70 millivolt. You have to remember this because maximum time it comes in the exam. What is the amount of resting potential? So you have to answer that is a minus 70 millivolt. And in this condition, the potassium ion is 24 times higher inside as compared to the sodium ions. Okay. Then whenever the stimuli is converted to the nervous person, that is the neurons, then another potential starts, that is called as your axon potential. If the stimuli is very rapid, that axon potential is called as your spike potential. So spike potential, you no need to confuse. Spike potential is it's the another name of the axon potential. When rapid stimuli or rapid impulse flow is there, then that is called as your spike potential. Here in that case, sodium and potassium channel opens, more number of sodium ion enters inside, and inside becomes positively charged and outside becomes negatively charged. You have to remember this statement. In this condition of the action potential, the outside negative charge and inside is positive charge, and this is called as your depolarization. And that is the volume of the depolarization. If you measure it, it will comes in the 80 to 100 millivolt plus 80 to 100 millivolt, and it will last for 1 to 2 milliseconds. You have to remember this time. I saw it in some exam, some questions, so you have to remember the time. Then that if you look at the action potential, there are two phases, you can say. One is your rising phase. Rising phase means it is the sudden convey of the potential, sudden convey of the impulses. So there is the rising phase, and after getting a particular limit, after getting means 80 to 100 millivolt, it will again fall. That is called as your falling phase. So action potential consists of your two phase, that is your rising phase, and another is the falling phase. Then there is the repolarization. This falling phase is called as your repolarization. That indicates the nervous system again, the neuron which is depolarized earlier, it is again coming back to its original state. That means the resting state. That is called as a repolarization. And the potential for the repolarization is plus 35 millivolt. Here in this condition, sodium gets closed and potassium gets open. You have to remember this condition. Sodium gets closed and potassium gets open. Earlier in resting potential, there is both sodium and potassium gates are closed. In action potential or depolarized condition, the sodium and potassium gates opens. In repolarized condition, which is also called as a falling phase, in this case, sodium gets closed and potassium gets open. So we will understand this whole process in the form of this graph. This is the resting potential. It is minus 70 millivolt. In this case, the maximum inside the nervous system, that is your negatively charged, whereas outside nervous system, that is your positively charged. Then whenever a stimuli is converted to the neurons, there is sudden increase of the potential. It will increase up to 80 to 90 or 80 to 100 millivolt. That phase is called as your rising phase, and then after getting a particular limit, it will start to fall. It will try to come to its original state. That is called as your falling phase, or that is also called as your repolarization. Repolarization repolarization And this repolarization, generally it is start at 35 millivolt. Okay, and particular in this repolarization condition, this sodium gets closed whereas potassium get open. So again, there are two terms we are getting: positive after potential and negative after potential. Remind this both. Look, this is minus condition, this is minus 70 millivolt, and this both positive after potential, negative after potential, the potential is in minus in nature. Then what is the difference? Due to sudden opening of this potassium channel, means due to opening of this potassium channel, maximum number of potassium will come inside. So as a result, the inside becomes negatively charged heavily, and the potential will come down as compared to the resting potential. That's why it is told as your positive after potential. It is somehow little less as compared to the resting potential. Then after entering of the maximum number of potassium ion, now it will move little bit up, and the potential will become little upper than the resting potential. That is called as your negative potential, and then it will come to its normal condition, that is your resting potential. Okay, so these are the terms you have to get acquainted with it before going to the chemical conduction. I will summarize the terms again. You have to remember the resting potential, which is also called as your membrane potential. The value of the membrane potential is minus 70 millivolt, and in case of membrane potential, the inside is negatively charged and outside is positively charged. Then the action potential, which is also called as a spike potential, that is called as the stage is called as a depolarization. In this case, the inside is positively charged and the outside is negatively charged. And in this case, sodium and potassium ion gate is opened. Then in axon potential there are two type of phases there, rising phase and falling phase. Falling phase is called as your repolarization phase. Here the potential is 35 millivolt, and in this case sodium channel closed but potassium channel is open. Then there are two terms, one is positive after potential and negative after potential. Due to rapid opening of the potassium channel, more number of potassium will come inside, so it will increases heavy negative charge inside the neurons. So that's why it is called as a positive after potential, and its value is little less as compared to the resting potential. And due to entering the more number of potential, there is means I can say there is the crowd of potassium ion on the outside of the neural membrane. So it is means it happens the slow conduction of the potassium again inside the neurons. So that's why the potential little move forward. It is called as your negative after potential, and again the neuron will come to its original state, that is called as your resting potential. Okay, so this is the electric transmission of the nerve impulse. Then we will study about the that is your chemical conduction of the nerve impulse. So there are several chemical transmitter, like we studied, that is the acetylcholine is there, L-glutamate is there, GABA is there, gamma aminobutyric acid. It's the full form of the GABA. These are the several chemical transmitter which mainly transmits the messages from one neurons to another neurons. Okay. First point is insect nervous system is cholinergic. Why cholinergic? Because in acetylcholine is mainly acts as the neurotransmitter in insect nervous system. Then another type of neurotransmitter, that is your GABA. There are two type of neurotransmitter. One is your excitatory, another is your inhibitory. Inhibitory. So excitatory nerve impulse, it will help in the smooth conduction of the nerve impulse, and inhibitory nerve conduction, it will stop the nerve impulse. It will not pass the nerve impulse in the furtherwards. Okay, so GABA is one of the inhibitory neurotransmitter, you can say, and L-glutamate is again another type of excitatory neurotransmitter. Then some synapses generally use norepinephrine or epinephrine for the neurotransmitter, and that type of synapse is called as an adrenergic. Okay, so if in the synapse acetylcholine is there for the transmission of the nerve impulse, that is called as a cholinergic. Instead of acetylcholine, if adrenaline or epinephrine epinephrine is present, that is called as your adrenergic type of neuron. So you understand this diagram? This diagram is there, but I will draw this diagram again. So this is the axon terminal. This is another terminal. So this portion is called as your synapse. We have seen previously. This membrane is called as your presynaptic membrane. This membrane is called as your postsynaptic membrane. When the electrical impulse reaches up to the axon terminal, it will enter into the axon terminal or presynaptic membrane. Here inside the presynaptic membrane, so many vesicles are there. These vesicles it will come and it will reach up to the top portion of the presynaptic membrane, and these vesicles generally burst and it will secrete the acetylcholine. This acetylcholine transmits the nerve impulse to here postsynaptic membrane. On the postsynaptic membrane, so many receptors are present. This acetylcholine come and binds with the receptor. Those are present in the postsynaptic membrane. This is the chemical transmission. So I will summarize once. When the electrical impulse that passes through the length of the axon, it will reach up to the end of the axon. Once it is reached up to the end of the axon, it will make so many vesicles. Those are present in the presynaptic cleft. It will pass up to the membrane, and this vesicles generally burst and it will secrete the acetylcholine. This acetylcholine now binds with the receptor. Those are present in the postsynaptic postsynaptic membrane, and in this way the impulse generally transmitted. Here in this condition, maximum, if you see, maximum insecticides like organophosphate, organochlorines, neonicotinoids, these are mainly targets this portion, that mainly either generally they deactivate the enzyme acetylcholine esterase, or generally they mimic means they looks like mimic this, that looks like the structure, looks like the structure of the receptors those are present in the postsynaptic membranes. So in this way generally they deactivate the nerve transmission in the insects. So this is all about the nervous system, where we studied about the basic structure of the neurons. Then we studied about the nerve transmission like electrical transmission and chemical transmissions. So in this system, in this topic, if you have any type of doubt, you just please make it confirm and please ask me in the WhatsApp number. My number is given in the slide, and please connected with the AgriAddict and get updated with the AgriAddict. We are frequently trying for your better future. So please be connected with us. Thank you so much.